All right, Burton is online. Kinder is online. Here we go. Fisher is online. Hughes is online. Julia is online. Let's see. Abigail here. Kincaid is here. Andrea. Is it Andrea? No, it's not. Mullins is here. Lewis. Lewis is here. Cooper? Not yet. Hunter is here. Melton is here. Crowder is here with WVSU gear. Actually, so is Hunter. Reinhardt is here. The other Melton is here. Rogers. Talbert. Kennedy is here. Hannah. Larissa is here. Madeline is here, right? Yep. Hughes is online. Galloway. Cook is here. Durgan is here. Let's see. Samuel. Tate, right? Yep. Burton's online. Kinder's online. Jones. Canterbury. Tristan. Hobson. Ray. And Fisher is online. We've still got 2 minutes. Let's just do reminders. So don't forget your exam is due by Friday at 9 a.m. So make sure you get that done. You want help with the study guides before that, let me know. What else? You should have 40 points of independent work by the end of, or by Sunday. You definitely want to work on that. I know at least one person's completely done, and some people haven't even started. I'm telling you, at least write a paper. I've been challenging you from the beginning. Write a paper. Think of a biology question, write a paper. Whatever it is, whatever you can think of that's a biology question, think of it, answer it. Because some of you know, even like the one who was done really soon, really early, they had issues, right? And you don't have to admit who you are if you don't want, but there was issues with some of their papers. So you might think, oh, I'm just going to turn this paper in and get 10 points because it's a paper. Like, no, it's per line, and you get points deducted for this, and you have to have your sources right for that, right? You guys know. So for those of you who've been making mistakes, it's perfect because you're making these mistakes early. Some of you people aren't going to turn your stuff in until the last day that it's due and realize that you don't have enough points because you didn't realize, oh, I have to do my sources this way, and I can't copy, and this is technically plagiarism, right? Can you guys agree, some of the who've turned papers in and had some issues with them, right? Because that's how you learn. It's a learning process. You turn it in, make a mistake, and learn from it. But you don't want to make a mistake right before it's due and then not have time to fix it. Had somebody come in. Hold on, I'm going to guess it, though. I know this. Canterbury, right? Yeah. Speaking of which, am I missing anybody for attendance? Everybody's been called. All right, it's 8. So the other reminder, too, is labs. You guys be careful with labs. I told you we've kind of been easing into it, so now it's strict. You have to have your pre-lab done before you come to lab, including any videos that might be on there. So for this lab, for example, there was a thing at the beginning of the introduction that said, hey, watch this video and do these questions before you read the rest of the pre-lab. Then right before you got to the questions, the pre-lab questions, it said, hey, before you answer these questions, make sure you go watch that video first. So it's all for a reason, and I know it sounds strict. For those of you who are on the Monday section, you're good, because if you mess up, you can just show up on Wednesday. If you're in Wednesday section and you screw up, then you're just out of luck. So make sure you get your labs done, including the videos, pre-labs. And like I said, I'm going to be more strict on the pre-lab deadline. So if you turn in your regular lab on Saturday instead of Friday, I'm probably not going to care. You guys have probably noticed that. I'm pretty loose on those due dates. But the pre-labs are important because you need to know what you're doing before you come in. Anyway, any questions about that? All right. Let's get started since it's 8:01. Any questions about anything at all? All right. And I guess the other thing to remind you about, you have 50 questions to take, 50 minutes to take about 50 questions on the exam. So if you want more time, we should find a time to meet online. And as long as I can see you and hear you and see your screen and make sure that you're taking the open book exam, I don't care how long it takes you. So if you need more time, let me know. And that's it for announcements. If no one else has any questions, then we can jump back into this new chapter, chapter 3, where we are learning about cells. Where are we at here? Slideshow from current slide. There we go. Let's do the first word for attendance. Here we go. How about space? That'd be the first word for attendance. All right. So as I mentioned, it's been a while because remember Monday we met. Well, we had that voluntary meeting you didn't have to come to, which was the exam review. By the way, the video's been posted for that. But anyway, so we haven't talked about this since last week. It's been a bit. And I wanted to remind you that a lot of this stuff that your textbook talks about, we slide right past it. We're going to fly through it because you're really going to learn more about the cell, the parts of the cells when you learn the important things that they do. Like this, for example, you're going to learn about chloroplasts later when we talk about photosynthesis. You're going to learn more about the mitochondria later when we talk about respiration. You're going to learn more about the nucleus and the chromosomes later when we talked about cellular reproduction. So that's why for now, I'm going through these quickly. We'll come back to chloroplast, but for now, let's talk about it. That's where the photosynthesis happens. So again, you're going to learn all about it. They also have double membranes. That's nothing that necessarily I'm going to ask you about. You don't need to know that it has a double membrane. But later, when we start talking about how photosynthesis works, then the fact that it is double membrane, that'll be important then. Because it's the whole structure function thing. So you'll learn at that point why that's important. But for now, it's just a little factoid for me to give you. So any questions about chloroplast? And again, to remind you, big picture here, the last thing we left off talking about was these differences between plant cells and animal cells. And the first thing we mentioned was the fact that plant cells have walls. And then now here's the other thing, plant cells have chloroplast. So any questions about chloroplast? All right, one last thing. So you can see all these parts. There's the thylakoid, there's the stroma, there's the granum, there's the inner membrane, there's the outer membrane. At no point am I going to ask you the parts of a chloroplast. Later, when we start talking about how photosynthesis works, I'm going to use some of these words. I'm like, oh, and this thing happens in the thylakoid, and then this thing happens in between the membranes, right? So I'm going to be using those terms. At that point, you'll need to know them so you know what I'm talking about. But when you're studying, don't feel like you need to know the parts. There's not going to be a picture on the exam where I say, what's the name of this part? What's the name of that part? All right, the other thing that plant cells have, the animal cells don't have, is a central vacuole. And put a big old X through this, not because it's not important, but just because I'm not going to ask you about it on the exam. And once we're done talking about it now, we're barely going to talk about it again. We will mention a little bit about what happens when plants get dehydrated or when they have too much water, things like that. And when we do talk about that, we're going to talk about the central vacuole again, but there still won't be any questions about it. So any questions about this slide? All right, animal cells. Animal cells have an extracellular matrix. Again, this is one of those things where I'm not going to ask you any questions about it on the exam, but it's in your textbook, so I'm going to briefly talk about it. The extracellular matrix is basically the stuff outside of your cells. That's basically what you should think of when you think of that. It's mostly made of glycoproteins, which are stuff you don't even need to know for this exam, and we don't really cover it much, neither does your textbook. But you could look it up if you want for independent work. What exactly are glycoproteins? What about collagen, right? So your book tells you that collagen is secreted from the cell, but what is collagen? What does it do? It's not important, which is why I'm not going to talk about it, but if you want to look it up for independent work, you can. All this stuff holds cells together and also helps make tissue, meaning that's one of the ways cells are attached, right? So, you're not just a big blob, right? You're a bunch of attached cells, and that's one of the reasons why, because you have this extracellular matrix basically holding everything together. And obviously cells need to communicate with each other, and that's another thing that the extracellular matrix allows it to do. But again, no questions about this on the exam. Do you guys have any questions about this slide? Riding through. Here's another one. Put another X. One of these things I'm going to talk about briefly, but it won't ask you about on the exam. And we have something called intercellular junctions. They are the direct contact between cells for communication. So this is basically, as far as we're concerned, like a subcategory of the last thing we talked about, right? So we're talking about extracellular matrix. Well, within that is something called the intercellular junctions. They have words for them. Of course, they're going to be a little bit more complicated to make things more difficult, but plasmodesmata junctions between plant cells. Again, I'm not going to ask you that word. We'll never even talk about that again for the rest of the semester, which is why I find it unimportant. Anyway, if plant cells have that, animal cells have what's called tight and gap junctions in addition to the desmosomes. But again, you're probably like, this is just a bunch of words he's throwing at me. Don't worry about it, because this stuff's never going to come back for the rest of the semester. Any questions about that side? All right, here's all the pictures we're talking about. So you can see these connections between the cells, right? The plasmodesma. You see where the cell wall is in the cytoplasm. If I were to show you this picture, which I wouldn't, but based on everything I just showed you, I should be able to show you this picture and say, is this a plant cell or an animal cell? And this should stick out to you as a plant cell, because I'm talking about the cell wall. Or you can see here, there's the cell wall and the vacuoles. These are all things that we talked about. Anyway, we see the plasma membrane over there at the top right with those tight junction proteins keeping the cells together. You can see here we have a plasma membrane with some cilia coming out of it, right? You can see how they're connected. Anyway, again, these are just examples. For the rest of the semester, these are not important concepts. So any questions about this slide? All right. So that brings us to a little bit, this is more important. Finally, we're getting into the more important, as far as the exam is concerned anyway, the more important part of this chapter. So what this whole exam 2 basically is all going to be about energy, right? So this chapter about cells is almost all about, like, why do cells need energy, right? This is kind of like what we're figuring out in this chapter, why cells need energy. And then the 2 chapters after that, well, how do we get the energy? So keep that in mind. Everything, this is the big picture of this exam 2 is going to be about energy. So let's talk about the cell membrane, because that's part of why we need some energy. And the cell membrane, this is very relevant to what we're doing in lab this week, right? We're talking about diffusion and osmosis and all those things. So the learning objectives of this section are to understand the fluid mosaic model of the membranes. That's actually a little bit less important. We're going to go through that pretty quickly. But then describe the functions of the phospholipids, proteins, and carbohydrates in the membrane. So even that's a little bit less important. We're going to talk about it. I have to let you know what's important and what's unimportant as far as the exam and the rest of the semester is concerned. So here we go. The fluid mosaic model of the cell membrane. It describes the structure of the plasma membrane as a mosaic of fluid components. Meaning, earlier I told you when we were talking about the biological molecules, and I taught you about lipids, and one of the lipids I taught you about was the phospholipid, right? The phospholipid bilayer that makes up the cell membrane, and I told you that's what the cell membrane is, that it's mostly phospholipids, and it is, but now I'm getting a little bit more specific. Now I'm telling you it's not just those phospholipids. You can see in this picture there's other stuff in them, right? Here we have a glycoprotein sticking out of it. Here we have a peripheral membrane protein. Here we have an integral membrane protein, right? Here we have some cholesterol, which we'll talk about a little bit. Protein channels, we'll talk about those a little bit later. But again, for studying, this is not a picture you need to study. I'm not going to ask you any of these pieces. You do not need to be able to identify these by the way they look. And a lot of them we won't even talk about. Probably the more important one, as far as the exam is concerned, and understanding stuff after this chapter, would be these protein channels. So we will talk about things coming in and out of the cell, in which case that will be important. But a lot of this other stuff is not. So any questions about this slide? All right, I'm going to underline the next word for attendance. There it is. Is that too small for you guys? Everybody in the back can see that, right? Okay. So yeah, fluid mosaic model. It is a phospholipid bilayer, which is like you can almost think of it like an oily goo almost. But yes, there's other stuff in that phospholipid bilayer. Here we go. Animal cells have cholesterol in the membranes. Why? Well, it regulates fluidity, and it sort of serves as an antifreeze. I'm definitely not going to ask you those two things, and I'm probably not even going to ask you about the fact that animal cells have cholesterol in the membrane. Because again, some of the stuff when we really get into how cells work later, when we talk about photosynthesis and respiration and genetics and all that, we'll talk about all the important parts then. And this is just not one of those parts that'll come back. So any questions about this? All right. Like I mentioned earlier, the membrane is mostly made out of the phospholipid bilayer that we talked about earlier. And like I mentioned earlier, too, when we first started talking about that, the surface is hydrophilic and the inner part is hydrophobic. And you don't need to necessarily memorize which one is which, but it is important for you to keep that in the back of your mind that one part is hydrophobic and one part is hydrophilic. It doesn't even matter to me that you remember which is which. The point is, some of the stuff I'm going to teach you later about how things come in and out of the cell, this will be important. Because sometimes things cannot come in and out of the cell simply because they are hydrophobic or hydrophilic, and they just can't make it through this weird membrane. So that'll come back later, but I'm not going to ask you any questions about it. Do you have any questions about this slide? All right. The proteins are the second major component, because again, like we're still talking about this big fluid mosaic model, that picture you saw. Well, we're still talking about that, right? So we talked about the fact that it's mostly phospholipids, but the second major component is proteins. And I'm not going to ask you that, and it's not that important, but some of this stuff down here we will talk about. We will definitely talk about enzymes, which will be important. We'll talk about attachments for cytoskeletons, but that's not very important. Cell recognition for us, obviously there's very important things for life. But for this course, cell recognition isn't going to be that important. So basically those two ones are going to be the most important. Not necessarily for the exam, well, maybe even that too, but those two are the ones we're going to talk about more. We'll talk more about channels of pumps and enzymes. Those things will have more of a purpose for us, for the story that we're learning about cells. Any questions about this slide? All right. Again, still talking about the fluid mosaic model. The number one component is the phospholipid bilayer. The number two component is proteins. The third major component would be carbohydrates. Those carbohydrates are always found on the outside of the cell membrane, right? So it's not on the inside where the rest of the cell is. And it's always bound to lipids or proteins. So they're not just like hanging out there. These carbohydrates aren't just hanging out there by themselves. They're bound to lipids or proteins. And they are used for cell recognition. I'll put this big old X through this, reminding you that none of this will be on the exam, and we will not talk about this later in the semester either. Any questions about this slide? We're skipping through a lot, right? But I teach out of the textbook, so even if it's something I think you don't need to know, I do want to at least mention it. So if you're following along in the textbook, you can say, all right, you didn't skip it. Let's talk about something a little bit more important as far as the lab is concerned, which is passive transport. So, as you already know from the lab, hopefully, you've either done the lab and or done the pre-lab, but these cell membranes are selectively permeable, which means they control what comes in and out. So you definitely need to know what that term means. Not because I'm going to ask you, but because I'm going to use it a lot, right? So cell membranes are selectively permeable. Not just anything can come in and out. They need to be able to regulate. And as you learned from chapter 1, when we talked about these life functions, like what are some of the things that, these characteristics of life, one of them was the ability to control your environment, right? And that's exactly what a cell membrane does. It controls what's inside of it, the concentrations of stuff inside of it. Sometimes it needs to let things in, sometimes it needs to let things out. And that's a lot of what we're going to learn in this chapter and in the lab. Anyway, any questions so far about the selectively permeable membranes? All right, if you download the PowerPoint, you can watch this animation of it. If you download this PowerPoint, you can also watch this animation. Here we go. Here's stuff you should already know, probably because of lab. Just in case, we're going to cover it very quickly. As you learned in lab, it's something called Brownian motion. I don't remember if your textbook mentions it by name or not, but that's what we're talking about here, right? Molecules vibrate and wander randomly. These pictures are not from your textbook, but I like them. So this is a water molecule, and they're not just sitting there. They're moving around. First of all, they're moving around in space, right? So if there was water vapor in this room, those things would be moving around, right? They wouldn't be stationary. But even if they were stationary and not moving around this way, even then they're moving, right? So this one, the bonds are stretching, right? So this thing is going back and forth, right? It's stretching. This thing's doing the same thing, except now it's like doing it not in sync, right? So it's going like this. This one's kind of flapping like this, like it's flapping its wings. This one's bending. This one's twisting this way. This one's twisting that way, right? So there's all these different ways that molecules can move, right? And you have to imagine if they're right next to each other, they are hitting each other like little pool balls. Like imagine that thing does a little stretch like that, then it goes and hits that thing, and then it's going to go flying around and doing whatever it's doing. But yes, so try to picture that. Instead of thinking about this at the molecular level, just try to picture things actually that size. Maybe sitting on ice, right? They're hitting each other, they're bouncing around, they're bouncing off of each other, bouncing off the walls. This is what's happening with molecules. All right, some things you definitely need to know about diffusion that somehow people mess up a lot. I don't get this. To me, this is the most important and simple part about diffusion. We're going from an area of high concentration to low concentration. By definition, diffusion is going from high concentration to low concentration, not the other way around. Somebody will miss that. But it should be simple, right? Like when you light a scented candle, the scent eventually goes out through the room, right? It'll start heavy here, and then eventually they'll be able to smell it, then eventually they'll be able to smell it, then eventually wherever you walk in the room, it'll smell the same. Because it's gone from an area of high concentration, which is where the candle's burning, to an area of low concentration, which is the rest of the room. However, you cannot open the jar and have all the smell come back into the jar, right? That would be going from an area of low concentration to high concentration. That's not going to happen. So yes, definitely know that we go from high to low. Also know that diffusion goes until equilibrium is reached, which is what I just said earlier. I just didn't use those words. I said that the scent would be all around the room, right? That would be equilibrium. Equilibrium is reached when the concentration is completely equal. And because we're going from an area of high to low concentration, this does not require an input of energy. This is just nature does its thing. Diffusion just happens in nature without an input of energy. Because, again, those molecules are just bouncing around. They're bouncing off of each other. All right, so any questions so far about diffusion, that part of it? All right, now try to imagine separating pure water from a mixture of dye dissolved in the water, like this slide here, and that's a good picture. It's a good visualization of diffusion. If you could almost imagine in your head, because again, these are not stationary, they're just bouncing around, right? Randomly bouncing around, bouncing off this, bouncing off each other. Eventually, they come across the membrane, right? Because the membrane does let them through in that part. And they keep moving back and forth until equilibrium is reached. Now, this is important for the exam. This is good to keep in the back of your mind. When equilibrium is reached, then the net movement stops, right? Meaning, see right here, we have a really high concentration and a concentration of 0. Then as it's moving towards equilibrium, then we have a little bit lower of a concentration than it was, and a little bit higher concentration here than it was. Excuse me. Then when we hit equilibrium, this is equal, right? This concentration is the same as this. But what's really happening at the molecular level is those molecules are still moving. I said that there's no net movement, right? There's not, the concentration's not going to change anymore. It's now equal. So yes, some of those balls will keep going to the left, some will keep going to the right. But overall, we've hit equilibrium. So the overall number won't change. It would almost be like if you were talking about two countries that are living right next to each other. If the immigration, coming in was equal to the immigration going out, then the population for those two countries isn't going to change by much, right? It's going to go up, it's going to go down a little bit, but for the most part, equilibrium was reached. Any questions about this slide? All right. Now this picture is the same thing, except now we've just introduced another color, right? So same thing. We've got the blue balls on the left, the red ones on the right. They're bouncing around until equilibrium is reached. We went from an area of high concentration to an area of low concentration until equilibrium was reached, at which point there is no more net diffusion. All right. Any questions about this slide? What are some factors that influence the rate of diffusion? Well, the way your textbook writes it, as you can see, is slightly different than the way that's written in the lab, but concentration gradient, mass of the things that are being that are in the solution, the temperature, and the solvent density. So yes, I know it's slightly different, but for the exam, this is what you should know. As a matter of fact, to be honest, I'm probably not going to ask you these questions on the exam. I'd almost rather just focus on it in lab. Like you're learning it in lab, we'll just leave it in lab. But anyway, according to your textbook, those are the factors that influence the rate of diffusion. And we'll talk about them. Again, if you download the PowerPoint, you can see another animation. Here we go. So we're still talking about passive transport here, right? So under that big umbrella term of passive transport, we got specific and we were talking about diffusion, right? Diffusion was a type of passive transport. Now we're still talking about this. Now we're going to get a little bit more specific and talk about something called facilitated diffusion. As you might imagine, facilitated diffusion is a type of diffusion, which again is still a type of passive transport. So what is it? What causes it? Well, there are some substances that don't cross spontaneously through a membrane, and they have to be transported through these proteins. So you can see here on the left of this picture, whatever this purple molecule is, its size and its chemistry allows it to just come straight through the membrane, right? There's no problems, it can come straight through. And with these, for whatever reason, whether it be the size or the chemistry of these molecules, they cannot make it through the membrane. So they need like a little doorway, basically, right? They need a little passageway, and that's what those proteins are for. It'd almost be like, imagine having 200 people in this room. Well, what happened there? Okay. If we had 200 people in this room and the doors were shut and there was 0 people in the hallway, diffusion is going to make people want to naturally leave this room, right? You're going to want to leave. We're going to be crammed in here, but you can't, right? The doors are shut. Now, other things can come in and out of this room. Air can still make it through the cracks of the door, but we are too big to make it through the cracks in the door. So what do we need to do? We have to open the doors to let the diffusion of humans naturally occur, right? Well, same situation here. Some of these molecules, they just can't make it through, so you have to open the door, so to speak. And that's, again, just facilitated diffusion. You should think of this is still diffusion, except it's specific to these molecules that just can't make it through the membrane, so they need a passageway, and the passageways are proteins. So make sure you know that. We're still going from an area of high concentration to low concentration. It still does not require an input of energy. This is still diffusion. This is still passive transport. Anyway, any questions about that? All right. That brings us to the next term that you're probably at least a little bit familiar with. Osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. So once again, we're still just talking about diffusion and passive transport, but this is yet another subcategory of diffusion, if you want to think about it that way, right? So this is a type of diffusion that's really specific. It's the diffusion of water. Not only is it the diffusion of water, but it's the diffusion of water across a selectively permeable membrane. And that brings us to the second bullet point, which we talked about earlier, and I told you it would come back again, and here it is. So these are terms you definitely need to know. might be asking them on an exam, but I'm definitely going to be using them. A solute is a substance that is dissolved in a solvent, and the resulting mixture is called a solution. So when you mix sugar with boiling water to make simple syrup, the sugar is the solute, boiling water is the solvent, and the simple syrup is the solution. If you mix salt and water to make salt water, the salt is the solute, the water is the solvent, and the salt water is the solution. That's why, I don't find these memes. I've not said that before, bro. I'll remind you. Or I find them, I don't make them. But I thought that was a good one. Because I see some version of this all the time, and that's not osmosis. Yeah, that's diffusion. Because it's going from, where the knowledge is going from an area of high concentration, which is the books, to low concentration, which is the brain. That would be diffusion, right? Not osmosis, because it's not water. Anyway, any questions about this slide? Okay. This is what I tried to draw in lab on Monday. So for those of you who are in lab on Monday, this will be a review. So you should, hopefully somebody can get this answer since you've already seen it. Let's forget that we're talking about osmosis for a second. Forget that it's low concentration, high concentration. Forget all that right now. Or for now, I mean. And forget too that this is a selectively permeable membrane. Imagine the green balls can travel back and forth without any problems. So in this picture, looking at that, completely forgetting about the fact that we're talking about osmosis and imagining those balls could go left or right. Are those balls going to go from right to left, or are they going to go from left to right? What do you guys think? Which direction would they go if we're talking about diffusion? Which direction would those balls go? Right to left or left to right? Right to left, good, right? Because like I said, in this situation, I said, let's pretend like we're talking about diffusion. So there's a really high concentration of green balls on that side, a lower concentration of green balls on this side. So nature would want to take its place, and the green balls would want to move from right to left. However, that can't happen. So now let's talk about reality, forget that imaginary situation. Now I'll tell you that these things, that little yellow dot, that is a selectively permeable membrane, and it does not allow the green balls to move. So equilibrium cannot be reached by the green balls diffusing. Instead, the water has to do it. So the water is going to go from an area where it's high concentration to an area of low concentration. Right? So the water will make its way to the right. Since the balls can't go to the left, the water goes to the right. I hope that makes sense. So the water is always going to go to the area that has a higher solute concentration, right? Because it's trying to dissolve whatever that solute is, or the solution. See right now, this has a really high concentration. This has a really low concentration. The water moves. As the water is moving that way, the concentration of whatever this green stuff is, it becomes higher, right? Because you're having less water in it. And here, the concentration of those little green things is getting lower because you're putting more water into it. And it just keeps doing that until it's reached, equilibrium is reached, right? So here you have more water than you do here, but the concentration is the same. You can tell by looking that the water to ball ratio is equal. So that's osmosis, right? So again, osmosis, we're still just talking about the diffusion of water. But the idea here is it's the reason that the water is moving is because it's still trying to reach a concentration gradient, or an equilibrium, right? We're still trying to equalize these two different solutions. There are some test questions, there are some study guide questions about this, and they'll probably be on the test. It's things like, and we'll talk about it later, but if you just stick, you have 3 balloons and 3 beakers, and each beaker is a 10% solution, and each balloon has a different concentration. You have to decide which one's going to be bigger at the end of it. I might have time to give you an example. Sure. Again, there's a question like this on the study guide, and maybe I'll do it later. I can't. It's not going to let me draw. I'll do it in lab maybe. But anyway, you'll see the test question. The study guide question, and if it doesn't make sense to you, let me know and I can explain it. Any questions about this slide? All right, some more words you need to understand just because I'm going to use them. And I'll probably test you on this whole hypertonic, hypotonic, isotonic thing. But tonicity, it describes the amount of solute in a concentration. Now, that's the word I probably won't ask you. I'm not going to ask you, hey, what is tonicity? But we will ask you the subcategories. So you need to understand what these words mean. Hypertonic, hypotonic, and isotonic. And hopefully those prefixes give it away for you. Hyper means there's a high concentration of stuff, right? Hyper. If you have, I don't know, if you say a kid's too hyper, what usually that means is like that kid has too much energy, right? It's a lot of extra energy bouncing around, jumping off the walls, right? If you have hypertension, you have high blood pressure, right? So when you think of hyper, you should think of high. And in this case, we're talking about the solute concentration. So if something is hypertonic, it has a higher concentration of solute. So again, if you had a test question, I'm going to try to draw this. I'll try to simplify it too. I'll say, no, that looks like garbage. Anyway, we've got one big beaker, and there's a balloon in that beaker. That balloon, let's say it has, I don't know, 10% salt solution. Wow, that looks like such garbage. Anyway, the balloon has a 10% salt solution. The beaker has a 20% salt solution. In this case, you would say that the beaker is hypertonic to the balloon, right? Because the beaker has a higher solute concentration. It has 20, the balloon has 10. So the opposite of hypertonic is hypotonic. That's when it has a lower concentration. So again, using that horrible picture, the balloon is hypotonic to the beaker, right? So here I am describing the same situation and using both words because it all depends on how you're comparing it. In this case, I went from comparing the beaker to the balloon and then the balloon to the beaker. So yes, hyper is higher, hypo is lower, and then you have the one in the middle, isotonic, right? That's when it's equal. So that's not in that picture. That's not isotonic in that picture that I attempted to draw. So this is the third word you've seen with that prefix, and you need to know all of them, right? Like an isomer is something that has the same chemical formula but a different shape, right? When we talked about fructose versus glucose. What's the other one we learned about? Isotope, right? So it's the same element, but it has a different number of neutrons, right? So iso, you should think of same. In this case, the same is the concentration. Any questions about these three slides? All right. To survive a hypotonic or hypertonic environment, organisms need to balance their uptake and the loss of water, right? And every organism is different. But that's what osmoregulation is. It controls the water balance. And that is actually pretty important, especially for aquatic animals. Like a lot of freshwater fish, they will die if you put them in salt water. Matter of fact, that might be an interesting independent work topic. You know, which freshwater fish can survive salt water, and how long can they survive it, and how, you know, what's the concentration of the salt water? Like, maybe they can handle brackish water, but not fully salt water. Another good thing to look up for independent work would be the hypothetical Noah's Ark situation. The whole world was flooded with salt water. What happened to all the freshwater fish? Anyway, osmoregulation. Again, all these organisms, this goes back to that first chapter, right? These characteristics of life. One of the characteristics of life is that homeostasis, right? Maintaining your environment. In this case, the environment we're talking about is how much water, right? How much solute concentration. And that brings us to some other words. Let's see. Isotonic, we already know. So if we're talking about animal cells, yeah, we are talking about animal cells. So yeah, this is stuff you should know. I'm glad I slowed down for a second, thought about this. So you will need to know the difference between animal cells and plant cells regarding osmoregulation. In other words, you should know what an animal cell likes, and you should know what a plant cell likes. So now you know animal cells prefer, generally speaking, this isn't always true, but generally speaking, as far as we're concerned, animal cells prefer an isotonic environment, right? So the amount of water coming in and out is equal. Yes, water's coming in, but the same amount is also going out at any given time. So it's not higher concentration, it's not lower concentration, it's equal concentration. So what happens if an animal cell is in a hypertonic environment? So again, let's imagine this is the animal cell, and I don't know, whatever solute we're talking about, let's say it's, I don't know, 50%, whatever it is, whatever solute we're talking about, it's 50% concentration. But its environment, this water that it's in, let's call it 80%. It's in a hypertonic environment. So what's going to happen, like I've said, the water is going to try to equalize things. So the water is going to leave this cell and go into the environment because the environment's a lot higher concentration, right? So it wants to even things out. And that's why a hypertonic environment dehydrates an animal cell, because the water is just leaving. The opposite is true if it's in a hypotonic environment. So instead of this, if the numbers were flopped, right? If the cell was 80% and the surrounding environment was 50%, then the water would be going into the cell. And that's not a good thing for animal cells because it'll make them blow up, basically, which is what this word is, lyse. So if there's too much water coming in, it'll just blow the cell up. So anyway, the next slide I'm going to show you is a picture of all this, and hopefully it'll make more sense, at which point I'll ask you if you have any questions. But there we go. See, in a hypertonic environment. Oh, sorry. Now we're talking about plant cells. Sorry. Let me back up 1 second really quickly. Make sure you understand this. So this is all about animal cells. And you need to know, again, isotonic is preferred. Are there any questions about this slide? All right. So really quickly, plant cells are different. One of the reasons they're different, as you know, is that plant cells have a cell wall. Because of that, they can expand, right? They can take water coming in. So because of that, they prefer to be in a hypotonic environment. Because in a hypotonic environment, water is going to be coming in. And that's what gives plants its structure. Well, that and cellulose, but right, those cell walls get filled with water. Those cells get filled with water. And that's what makes a plant turgid. And that's why when a plant gets dry, it gets all wilted. Because it's in a hypertonic environment, water's left. So there you go. What do animal cells prefer? Animal cells prefer isotonic. What do plant cells prefer? Plant cells prefer hypotonic. So animal cells like an equal amount of water. Plant cells want water coming in. If you can just remember that plant cells have plant cell walls and the big old vacuum that holds water, hopefully that can help you remember that plants like to have water coming in. Anyway, any questions about this slide? We're making pretty good time. As I've already mentioned, here it is again. I can see a picture of it. As a plant cell loses water, it shrivels, and the plasma membrane pulls away from the cell wall, and that usually kills the cell. So this one's probably not too dead yet. I have a video somewhere where I took a, what's that called? Not stop motion, but time-lapse video. Somebody failed to water my plant at my house, and it did look like that. So I watered it and then videotaped it or recorded it for like, I don't know, an hour. And of course, that hour turned into a 30-second video. But you could see it go from this to this, all because it was watered. Anyway, any questions about this slide? Again, hypotonic environment is best for plants because plants like to have that water coming into the cell. And then here it is all at once, right? This is not a picture from your textbook. This is a picture from your old textbook. It's a great picture that shows them both. Animal cells versus plant cells. Animal cells prefer isotonic. Isotonic's not great for plant cells. Plant cells prefer hypotonic because they have all that water coming in. That's great for them. That's horrible for animal cells because they'll just blow up. And then hypertonic is bad for both of them because regardless, the water's leaving the cells and the cells are becoming dehydrated. So any questions about this slide? Which brings us to the last bullet point of this chapter, which is great. Maybe Friday we can actually start the next chapter. We'll see. Active transport. So again, remember, we just finished talking about passive transport, which was diffusion. So wouldn't you guess, actually, how many of you heard active transport before? Just a couple of you, which makes sense, right? Because it's not one of those common terms you hear unless you're in biology. But even for those of you who haven't heard active transport, you probably can imagine that active transport sounds opposite of passive transport. And basically it is. So keep that in the back of your mind and we'll talk about what it is. The learning objectives of this section are to understand how electrochemical gradients affect ions. That's what your textbook says. We'll mention that briefly, but you don't need to know that. You'll need to be able to describe endocytosis, including phagocytosis, pinocytosis, and receptor-mediated endocytosis. And I might ask a question or two about that. But even then, it's not really important because most of these concepts are not important for understanding concepts later in the semester. And then finally, understand the process of exocytosis, which is just about as important as that bullet point. So we'll go through this pretty quickly. Active transport, unlike diffusion, it does require an input of energy, right? So diffusion or passive transport does not require an input of energy, but active transport does. So you need to know that active transport requires energy. It also, when you ask what kind of energy, where does it get its energy? There's something else you need to know, ATP. Very important. I'll circle it. That'll be the next attendance word. ATP. If you forget everything else in this chapter, remember this one fact. Cells need ATP for energy. Because that's what most of this exam 2 is going to be about energy. The whole thing's about energy. Chapter 5 is basically saying that one thing. Cells require ATP for energy. Then the next chapter is going to be like, well, where do we get ATP from? And the answer is from respiration, because we break down glucose to make ATP. And then, by the way, the next question will be, okay, then where do we get glucose? And that answer is photosynthesis. So by the end of this chapter, it's kind of like we're working backwards. First, I'm going to tell you that we need ATP. And then on the next chapter, I'm going to tell you, all right, we make ATP by breaking down glucose. And then chapter after that, I say, okay, and this is how we make glucose. Anyway, active transport also requires a transport protein, which almost makes sense, right? Because you're going to need something. If you require energy, it makes sense that you would need some sort of protein for that. Again, just like the opposite of passive transport or diffusion, it moves solute against the concentration gradient. So here we're going from an area of low concentration to an area of high concentration. And why would you need to do that? Well, your cells just need stuff, right? Sometimes your cells maintains the concentration of small solutes that differ from the environmental concentrations. So like I said earlier, we need glucose to make ATP, right? So your body, your cells need glucose in them. And I bet, well, I don't know this for sure, but this is a good example just to picture it in your mind. Imagine, your cells have a concentration of, 70% glucose solute, but your blood is only, 60%, right? So then your blood has less, or is hypotonic, but yet we still want to have more of it in, right? We still want to bring that, the glucose in against the concentration gradient. And that's what active transport is used for. Sometimes you just need stuff, basically. Any questions about this slide? All right. Electrochemical gradient. I'm really only going to talk about this briefly because this, again, is one of those concepts that if you don't remember it, you'll be fine because you don't need to understand this to understand the stuff I'm going to teach you later in the semester. But your book does point out that the interior cell is negative relative to the exterior. So I mentioned earlier about that whole semi-permeable membrane. I'm like, sometimes something can't come in and out because of its size. Sometimes it can't come out because of its chemistry. And this is just coming back to that. That's what I mean by its chemistry, right? One of the things to look at is the fact that, again, the inside is relatively negative compared to the outside. And of course, if you're a charged molecule, that's going to make a difference, right? So not only do we have the old hydrophilic thing that we talked about earlier, but now I'm also telling you there's a difference in charges. So long story short, there are reasons other than the concentration gradient that necessitate active transport. So you don't need to know any of this. The point is that, yes, when you think of active transport and the fact that we need to put energy in, it's, as far as we're concerned, yes, it's because we're going from an area of low concentration to high concentration. That is an acceptable thing to think, but also just for your own mind, technically, there's also other reasons, right? It's also this electrochemical gradient. And that's the last we'll talk of it. We will mention hydrogen ions later. Hydrogen ions are positive, so that is technically that's involved with the electrochemical gradient. But even then, I won't even bring up electrochemical gradient anymore. So any questions about this slide? Here's some pictures, or excuse me, if you download the PowerPoint, you can watch this animation. So you'll be able to see some things coming in, some things coming out. This is what's called a sodium ion pump, sodium potassium pump, but you don't need to know anything that specific about it, but you can see it in action. But here's a picture of what we're talking about, sort of. So there's the outside of the cell on the top. Here's the inside of the cell on the bottom. Here's the phospholipid bilayer. There's this protein that we're going to use for this active transport, right? So you can see there's a low concentration of whatever solute that is out there. It's a higher concentration inside, yet the cell still needs more of it. So it's going to require energy. You cannot just diffuse it in because it's going against the concentration gradient. So where does it get its energy from? It gets it from the ATP, right? ATP activates it. That gives it its energy, and then it can then pump that solute against its concentration gradient. And this will be an important concept. When we start learning about photosynthesis and respiration, those two things wouldn't happen without this process right here. Without something going against its concentration gradient, then we couldn't have photosynthesis and respiration. But without that, we just couldn't have life. So as simple as this is, and as few questions as there will be about this on the exam, it is a really important concept. And also, I think it's funny, too, that the book does correctly mention that usually it's ATP that gives it the energy to do this. But the two times we're going to talk about it for photosynthesis and respiration, the energy comes from somewhere else. And that will be important when we talk about it. But for now, I just want to point that out. Any questions about this slide? All right. So now we're done talking about active transport, right? We talked about passive transport. We talked about active transport. Now there's yet another way that things come in and out of cells. And that's what this is, endocytosis. Yeah, I forget. Technically, it is an active transport, technically, because it does require an input of energy. But this is when cells take in material via vesicles. So remember when we first started, I taught you about vesicles earlier. I said, yeah, we're going to hear this word again, but it's not going to be very important. So here it is again. You're hearing the word again. Vesicles is just like little tiny pieces of the cell, right? So try to picture what's happening here. All the solutes coming to the cell membrane, it kind of buds inward until it pinches off, and now you have your own little bubble of cell membrane with the solute on the inside. So the easy part about this, and it's going to make me sad when someone misses it, not because it's important, but because this is going to be a test question, and I always think it's an easy one. I like to put some easy ones in there. Endocytosis is stuff coming in, right? Just keep that in the back. Keep that in your mind. Endocytosis, stuff coming in. It's almost in the name, right? Endo. Of course, it's spelled differently, but phonetically, that's the same, right? It's when stuff's coming in. What do you think the opposite is called when stuff leaves the cell? It's not on the slide, but if you had to guess, if endocytosis is stuff coming in, what do you think stuff coming out would be? She said it. But yeah, exocytosis, right? And we'll talk about that in a second. So at least remember that. Those questions will be on there, and I'm putting them on there because I think they're easy. Where it does get a little bit more concentration, complicated, is endocytosis is then subcategorized into two different versions. You have phagocytosis, which is basically cellular eating, and pinocytosis, which is basically cellular drinking. So if it's solid stuff that's coming in, that would be phagocytosis. If it's liquids, it's pinocytosis. The way I always remembered that is when I think of wine, right? Pinot Noir, Pinot Grigio, right? So when I think of the prefix Pinot, I think of a wine, and I remember that wine is a liquid, so that's what helps me remember that. Anyway, any questions about this slide? All right, we already talked about this one. The opposite of endocytosis is exocytosis. Just when stuff leaves the cell, right? So the vesicle with the stuff in it merges with the membrane and just puts it out there. Any questions about that slide? If you download the PowerPoint, you can watch this little animation of pinocytosis. Wow, we did it. All right, the last word for attendance is right there on the screen then. Discard is the last word for attendance. If you haven't done your lab, like if you're just to come to lab today, make sure you do your pre-lab and your video. And let me know if you need help. That's it. Otherwise, I'll see you guys in lab, or I will see you on Friday. If you came in late, make sure you email me and remind me that you came in late.
I can't do it.
Yeah, sorry. Yeah, please. I appreciate it.
Sign this.
Yes, I need to take it up to my office so I can look